free discussion. Feel free to ask questions. And we're going to cover the full workflow. First, I'll just give you a brief biological background, and so that we're all on the same page. First of all, I would like to acknowledge my collaborators, Frank Paulo from Columbia and Yusuke from University of Tokyo. We really wanted to study ER, mitochondrial contact sites in neurons, but with all the complications that comes from fib milling and working with neurons, we just decided to work with models, mammalian cell systems like HeLa cells and NIH cells. But the research has broader implications for neurodegenerative diseases, and these pictures of synapses are gonna make sense in a second. So this is really from another talk, but initially we have some biological background and contact sites between organelles. Some of you are actually working on that. Why do cryotomography and these things? And I'll show you my results. So if you look at the mammalian cell, you have many organelles. ER really stretches all over the cell, and it's really a powerhouse. It creates proteins, lipids, it stores calcium. So all the other organelles depend on ER for these things and many more things. So this is satisfied through contact sites, and the number of different kinds of contact sites is very vast and heterogeneous. ER mitochondria, ER lysosomes, ER plasma membrane, and then all the other organelles together. So it's really an interesting landscape, and these are, we can think of them as synapses inside cells where two membranes come together. The contact is fortified by tethers, complex protein complexes that hold them near each other, and there's going to be some exchange of biological molecules or ions and etc. So it's a very hot field now in biology, biochemistry, and it's a very interesting thing to study with cryo-EM because again, the physiology is very vast, so you can look at the same thing like ER mitochondrial contact sites in many different contexts and get completely different tomograms and completely different structures. So it's very exciting. And these are just different examples of what can happen at these contact sites. Lipid transfer, like I said, is one of the important ones. Calcium signaling between ER and mitochondria, and fission or fusion of mitochondria with the help of ER are some of the examples. So this is what I wanted to study. Like I said, there are tethering complexes. The list is very long of all the candidate protein complexes that could be at these contact sites. And the list is probably longer and more complex. Here, you're only looking at an ER candidate and a mitochondria candidate that come together and form a tether. But the stoichiometries are not known, and you could have multiple of these proteins interacting at the same time. So one protein we are looking at is PDZD8. This one is the ER side of the tether. It has a single transmembrane domain and a bunch of different domains. It's actually all over ER, so it has promiscuous interactions. So it will probably have many different conformations, and this is like an AlphaFold prediction. You can see there's a lot of disordered regions and a bunch of familiar domains. And so we really don't know the structure of the full length. It's difficult to purify. It's difficult to reconstitute these contact sites in vitro. So that's why we chose to do tomography. And the other protein is FKBP8. I won't get into it a lot, but it also has a transmembrane domain that binds it to mitochondria. It's been studied to be involved in autophagy, etc. I'm just going to pass through these slides to get to the methods part. So what I wanted to achieve, and the result you can see here was, to be able to see these contact sites and also be able to get some kind of structure from the tethers that exist at these contact sites. So just to kind of give you an idea what the end result should look like. And just to remind you all of all the different things we've been talking about. We've been talking about putting cells on the grid. I'll show you a couple of slides about that, of the different things I tried, and you might be interested in trying what you can do with different FLM microscopes. So I'll show you examples of Zeiss Aeroscan and also the IFLM, which is the FLM module in Aculos. Fib milling, I think Deja has covered that really good. So I'll just show you some examples. And the tomography part, I don't have many slides on that, but this talk will also kind of be an introduction for our mirror session later. So I'll show you some segmentation results and discuss one specific workflow that Ben Barrett developed in San Diego. I'll show you some of my results from that workflow. So let's get into it. So I'm going to jump right into IFLM. Deja has probably brought it up. So this would be the FLM module inside Aculos. It's not coincident, so that introduces a lot of challenges. It's obviously more difficult to use than a coincident beam. And this is the Thermo Fisher version of it. There's also a Delmic version from that's called Meteor. And I got a demo of that in Rockefeller, and I actually like that one much better. But the reason would be the software was more streamlined and easier to use. So that's just a side note, but this is the kind of result you can get from the IFLM. So you can either look at your lamella after milling, and so here we have a reflection image at the top. So this is your lamella, this is the GIS front. These are the stress relief cuts. So this is not a waffle grid, just a plunge frozen grid. You can see the grid holes. And this is an overlay of the green channel, which has Tom-20 Venus signal in it on top of the reflection. So you can see where the mitochondria are in your lamella. And so mitochondria are easy to capture in lamella because they're all over the place. So that's why it looks really nice. But if you're looking at something more sparse, the signal will probably be weak or not visible with the IFLM. One interesting thing about IFLM is it has a large working distance. And when you compare it to Meteor, which has a much shorter working distance, it actually can limit what you can do. So I saw this poster in the tomography conference in Netherlands where they rotated the stage and were able to get FLM images of the lamella site in the milling angle. So these images are kind of like SEM angle, so it's like 90 degrees to the surface. But with Meteor, you can actually get milling angle images. So you don't have to do 3D transformations, and you know exactly where to mill, which is super useful. And this one is from a slice and view sample. So again, helicels, plunge frozen. You can see the boundary of the cell here. I've already done some milling, so that's what you're seeing here. And this is the puncta in the cell. And I'll show you, I think it comes after this. I was able to do slice and view and IFLM, like pause the slice and view process, get an IFLM image, go back, continue without having to do many adjustments, and go back and forth. So while you're getting a SEM volume, you can also get your FLM volume, which can be useful. So that's one thing you could do with these FLM modules. And this is just an example of a medium mag. It's not low mag, it's a medium mag montage of the same mitochondria from the signal here. So you have a long mitochondria here. This is before I started using glycerol. So you have these bright spots are from non-vitreous ice. You have the nuclear envelope here. So just an example. And this is the what you can get with slice and view with Aclos. It supercharges, and that's why people don't use it a lot. I have an example of the PlasmaFib paper if you want to see it, but it's basically the difference is between day and night, quite literally, because these are pretty dark because of the charging. And the PlasmaFib milling, you get super clear images. And this is the FLM stack. Not that many images, but you get the idea. Also, I did the FLM after the SEM imaging, and it turned out that the SEM imaging is actually killing the signal. So what you're looking at here is the signal around the imaged area. So what I wanted to try, but I didn't get to trying, is pausing the milling, getting the FLM, and then doing the SEM imaging, so that the damage doesn't kill the puncta. So that's that. All right, so now we're going to switch to Zeiss. This slide is about autofluorescence, which is something that people have talked about, but when I started doing this, I didn't know anything about it, and it was very confusing and difficult because it's a very annoying byproduct of doing, thank you, of doing cryogenic fluorescence microscopy. So in cryogenic temperatures, the decay time for autofluorescence is longer. So you actually, it ends up as a signal in your image. And you have it in all the channels. So with these first two rows, I'm showing that you can either get it in like this wavelength, detection wavelength, which is one on top of green channel. And over here, you have the actual signal. And this one is vice versa. So the signal should be here, but you also get a lot of signal here. So there's autofluorescence, there's leaking because some of the puncta overlay, but there's some puncta that are in the green channel that shouldn't be there. So that's what autofluorescence and leaking means. And this is a mixture of actually what I was interested to look at, which was look at PDZ-D8, my protein of interest, with a halo tag, not overexpressed. And then TOM20 Venus, which was overexpressed. So green is mitochondria pretty much. And the magenta is supposedly my protein, but we cannot say for sure because the intensities are very close to autofluorescence intensities. And that's one of the reasons we decided to overexpress our proteins so that we can differentiate between autofluorescence and actual signal. Sorry? So one of them is knocked in. Yeah, the halo tag is knocked in. And the other one was a plasmid, so a transient transfection. Yeah. 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 So yeah, that's a discussion that you're either going to have internally or with your collaborator. So in my case, the biologist was my collaborator, and we had this discussion back and forth. And you know, you want it to be native, so overexpression is not an option. But you want to do structural biology, so you need numbers. So I think the sooner you do overexpression, the sooner you'll get results. Otherwise, yeah, especially if you're milling. But this is all because you're going to end up with a tiny slice of your cell, and the signal is going to be weak. You're not going to have that many targets, so you have to overexpress.
I have a follow-up question. So
yeah.
Did you actually see them that they, mislocalize and sample somewhere besides the mitochondria?
Oh, not in this case, no. And that's partly because we introduced a mutation to the protein we eventually used as our guide, our beacon, and put the tag on it. we introduced a mutation that anchored it to only mitochondria. So I'm going to show you the fluorescence results from that, and it all overlays with mitochondria. So
Are you able to like where mitochondria eyes and mouth?
sorry, what's the
Are
question?
mitochondria, do they have eyes and mouth?
What do you mean? Eyes?
Yeah, I mean it's a living organ.
Yeah,
How does it see with their?
I'm not following the question. Sorry.
So mitochondria, how does it know where to go, like inside of the cell? Does it see it in it?
So so this might help you. It's all over the cell. So that's all mitochondria. So it forms a giant network inside the cell, and
But how?
it's very dynamic. So it moves around.
How does it sense? Does it have like? Are we? Do you have any like 3D image that you into inside of the mitochondria?
Yeah. I'll show you examples. Yeah. So it's very dynamic. So if you actually take a live imaging, like live fluorescence imaging of mitochondria, it's moving around. It's going through fission and fusion. So
Are you able to detect fission?
yeah. I think this example, which was way back here, that's not my work, but this is 3D EM volume imaging, probably room temp, of ER pinching mitochondria. This is from, I think, Gia Volz from University of Colorado. And
What's the function?
so ER brings in some proteins that help with pinching the membrane. So that's what ER is doing there. It just brings in the necessary machinery to pinch a membrane, which takes a lot of force. Well, they're going to form complexes. I'm not super familiar with and I don't. It's actually like an open question. So there are proteins identified that are involved in this process, but exactly we don't have a movie of this process with like molecular resolution.
Did anyone ever would detect like fission, like moving?
I mean, with fluorescence, yeah, and oh.
Like, I cannot even find a 3D image of mitochondria in the literature. Everyone's talking about maybe.
You should look into like 3D volume images, like Slice View or FIPSEM volumes. So basically, it's like milling. So you're milling the surface of your sample, and you take an image, and then you mill again, and you take another image. So you are going through the sample and taking images, and then you can construct a volume. And in that, you can see the full 3D structure of mitochondria. Yeah. Yeah, I can probably share some links with you. Yeah. So from this slice, mostly I wanted to show you that you can get amazing images with Zeiss. These are all 100x images. And the tags are all on mitochondria. So you should definitely try it. And again, more images of mitochondria tags. These are NIH cells. Okay, so this is the final results of how I decided to do all the overexpression stuff. So I'm overexpressing the mitochondrial partner, which is FKBP8. And on top, we have just the control, so no overexpression. And the tag is on PDZD8, and it's a venous tag. So the nice thing about venous tag, even in room temp, is that it's supposed to be a green tag, but you can also see it in the red channel. So what I'm showing here is actually the puncta that, for example, you see here or here, that's a better example, are not visible in the red channel. And so we use that as a clue that we're looking at autofluorescence here and not actual venous signal from the venous tag. And then after we overexpressed the protein, we got an increase in the intensities of all these puncta in the green channel, and accompanied by an increase in the number of puncta in the red channel. So that gave us some confidence that we're looking at actual signal. So eventually we put the tag on what we were overexpressing. So we put an M-scarlet tag on FKBP8. And the reason is we just figured out that red tags are just better. There's a lot of autofluorescence in the green channel. So whenever we want to look at what we're interested in, we put a red tag on it. So some overexpression phenotypes, some overlays. You can still avoid leaking and autofluorescence, but it's better than working with wild type. And these are some Zeiss examples from lamella. And so one thing I tried with my workflow was I did the FLM imaging after tomography data collection, and it didn't kill the signal, so that was a plus for me, but it's not always the case. So when you zoom on these, you'll see that the signal overlays with mitochondria pretty well. And so next slide would be about different ways of milling. So eventually I just worked with plunge frozen cells, but initially I was just curious to see, how can we increase throughput, and can we use high pressure freezing and stuff like that. And so these are just different ways of freezing cells. So plunge frozen here with lower concentration, plunge frozen with higher concentration, which usually results in bad ice, and high pressure freezing. And this is actually in combination with micropatterning. So all the cells are stuck to the square. So you have nothing on the grid bars. And that's what the surface looked like. So it works pretty nice, but you can have a very clumped or cramped situation with the cells. And that may not be ideal depending on your project. So, and it probably means they're not healthy, so it makes it a little tricky. But this is what you can expect. And since we are high pressure freezing and there's no blotting, you get ice all over your sample, and that you can see that effect in your lamella too. So this is where the film is. This was, I think, a carbon film. So you have a big chunk of your lamella is just buffer, and you have the cell on the other side. And these are just lamella examples of plunge frozen cells. Yeah. Like grow them on plastic and then put it on a grid? Is that what you're saying? Yeah. Yeah, the only complication is the shape of the grid bars. So, if it doesn't mess up how the freezing goes and how the surface looks at the end, why not? So ideally you want, I'm sure Deja's brought this up, but ideally you want, where's the mouse? There you go. Ideally you want a flat surface like this. So anything that prevents that is not a good approach. Yeah.
So why don't you just grow cell
on the other side? Yeah. It's just that experiment. I did it on this side. I think there was actually, this was a while ago, so if I put it on the other side, I would have gotten more ice on top of the cells. I think that was my rationale. Because the side that we usually do waffle on is 20 microns. and these cells can go up to 10 or 15 microns at most. So I would have this water layer in front of the lamella, which that's the prime real estate of your lamella, which, you know, the front is always the thinnest, cleanest part of the lamella. So I put it on the plunge freezing side to avoid that. Does that make sense? All right, so putting all of this together, you know, I eventually worked with HeLa cells with an M-scarlet tag on my protein overexpressed. This is a clump of cells. Some of them may not look super healthy, but anyways, I milled a lamella. This is a media mag montage of the lamella. And one thing to note is, even when you use glycerol, so this has like perfect ice over here, right? But you still get, with plunge freezing, you have a gradient of freezing. So there's some parts of the lamella that I cut out that had non-vitreous ice. But it doesn't mean that the rest of it is trash. And this is, the inset is the FLM image of this view. And so what I've pointed out with arrows are the contact sites that you can see with your eyes. So the reason for doing the high resolution montage was to be able to see these things easily. So we didn't even need the fluorescence for targeting. And that's another reason to do it after data collection. And these are the tomograms from these targets. And these are segmented and subtomogram average, so it's the final tomogram, not the initial tomogram, obviously. And so now I'm going to get more specific, dive a little deeper into the project. So I collected 70 or 100 TIL series for overexpression. 70 of them had contact sites. And with the control, I collected like 50 TIL series, and only 8 of them are useful. So another reason why overexpression is necessary. And I had to do all of the analysis manually, so like the subtomogram averaging, the segmentation. So I didn't process the full overexpression data, but I processed it to a point where, you know, it wouldn't make a difference with more data. So we'll get to segmentation first. So you're looking at an annotated voxel representation of your membranes, and you can see that, you know, the surface is really rugged, and if you try to do any quantifications, they're bound to give you meaningless numbers. So Ben Barr's workflow uses a screen Poisson mesh reconstruction, which means that it takes this voxel representation and generates a smoothed out point cloud. And so with this point cloud, you can do any kind of quantification you like curvature, distances. The opportunities are basically unlimited. And so what I was interested in, because I'm looking at contact sites, was the distance between ER and mitochondria. So I've mapped the distance on the ER membrane, and you can see the distribution of distance in these contact sites. So previously, people only looked at volume EM images, and you really cannot quantify this distance like we have done here. So that's like a big plus of doing high resolution tomography, is you can now zoom in and really characterize the distances. And instead of the like 30 nanometer distance that is in the literature with about the ER mitochondria distances, now we can see that it's actually more heterogeneous, and there's a lot more going on, basically.
Is it always the same part of mitochondria that
This is also dynamic, so they're constantly forming and disassembling. Yeah. And so these are, so the mammalian contact sites are huge. They can be microns long. So these are slices of the actual thing. But it's a trade-off between resolution and volume, right? So.
Did you ever do spectroscopy? It's higher
Good question. I'm not sure about iron, but it does regulate calcium. So, when ER releases calcium, if it happens at the contact site, it goes into mitochondria directly versus going to the cytosol, and that has like significant physiological differences.
Mitochondria probably doesn't like cost.
Yeah.
Probably different.
Yeah. I'm not sure exactly what the balance is like, when it goes in which way, but yeah. All right, so and then you can do aggregate analysis. So this is the ER mitochondrial distance plotted over all the tomograms between overexpression and control, and you can see there's a shift, which we think is due to the overexpression. And this is an example of how that looks like in one specific tomogram. So over here we have all the three membranes, ER, OMM, IMM, so outer mitochondrial membrane and inner mitochondrial membrane. And so this is like one example of a mitochondria, a volume image of a mitochondria. And so if you remove ER and look at the contact site, you can plot, all the distances. And you can do that for all of them and, gain interesting insights into the heterogeneity of these contact sites.
Is it opening and closing?
It doesn't alter. Yeah, it doesn't alter the integrity of the membrane. They're just like synapses. They come close to each other, but unlike synapses, I guess they can disassemble. But there's a lot of unanswered questions, so there might be some pore-forming mechanisms there. We don't know. Yeah. And you can also look at, you can quantify mitochondria itself by looking at the distance between the outer mitochondrial membrane and inner mitochondrial membrane. The hot spots is where you have cristae, and the reason is that the cristae, as you can see here, kind of look like holes in the inner mitochondrial membrane. And that means an increase in distance between the OMM and the IMM. So that's how you can actually quantify the cristae, which is very interesting. And you can compare it between overexpression and control. And what we saw was that the distances have a larger variance in overexpression regardless of where you're looking at. So either at contact sites or outside contact sites. In the overexpression, you have larger distances compared to control. And subtomogram averaging. So, if you look at the contact site, which I've highlighted here, on the right you have mitochondria, on the left we have ER, and in the middle, any density that connects them is potentially a tether. And you can see that it's pretty packed with different shapes and structures. Some of them branch and have legs, some of them are straight, some of them are like this. So there are many challenges with this. One is that you cannot subtract all the other signals, so that's going to affect the alignment. Also, you have 3 strong membrane signals in your subtomogram, which is also going to drive the alignment. So I was pretty sure that regular approaches wouldn't work, so I just jumped straight into doing it manually. But I tried to impose some, geometric information so that I can use it later to align them. And I was inspired by a method that Michael used from MRC. He used dipoles for yeast tethers, and the yeast tethers are pretty homogeneous. They all look like straight rods like this. But here I have a more heterogeneous landscape. You have tethers that look crazy, and even the straight ones have shorter ones and longer ones. So I decided to use 2 different dipoles, one that aligns with the longest density of the tether, so that will be the red dipole, and one that aligns with where the tether touches the membranes. And so in the case of a straight tether, you know, they align pretty well. And in the case of this tether, you can use the differences to classify them. And this is a simplified version of the full reality because, like you said, the shapes and structures are much more complex. And, you know, we can plot it and classify them, group them together. So I'm just going to skim past this. Yeah. So I picked in iMod, and what I did is just put points on either side of the tether, and then you can extract those coordinates. And yeah, I shouldn't have skipped this part. So you can extract the coordinates from iMod. So they're basically X, Y, Z of all the points. So iMod doesn't know that you're picking dipoles, but you can then import this into Dynamo, and with a little script, you can, and some dipole functions from Dynamo, you can turn these points into meaningful dipoles. And from there, Dynamo actually takes care of it. So when you extract them, it extracts them and calculates their Euler angles. So after you extract it and you average them together, they are all already aligned. And so without any alignment or classification, you actually get densities out of the tomogram. And so they're not biased, there's no reference to bias it. And you can see that I was able to capture some of the complexity of these tethers, but the resolution is low, obviously. And I think the greatest takeaway lesson from this is that the two interacting partner model doesn't really hold, because you can easily fit two proteins in the smallest one and still have some free space. And you can see that we have a large variety of sizes. So the stoichiometry should be figured out, so that we have to go back to the bench and do all of that stuff. And there's probably more than two interacting partners in each tether. And yeah. Mm-hmm. Sorry, what's that?
Like spiking it and stocking it in iMod also makes that directionality.
Oh, I don't know that. Is that a tool in iMod?
Yeah.
Okay. Oh, I didn't know about that. Probably the same. Yeah, all you need is like some directionality. Yeah.
And then it sets angles.
Yeah, and if you align them all to Z, basically the Z direction is your reference model. So you just align all of them to Z. Yeah. Yeah. Those are actually physiological holes, some of them at least. So they're actually all over the place, but that's actually a great point because, like I said, lipid transfer happens at ER contact sites. So to create these cristae, you would think that it should be close to a site of lipid biogenesis. So there's a hypothesis out there that maybe at the contact sites you have more cristae, but that's actually something I did not see in my data. So I saw a null effect. There was no really huge difference between contact site and outside contact site. But my system is altered. My mitochondria are mostly globular. I don't have many tubular mitochondria. So yeah. So and they're meaningful different types. Yeah. So the first example of mitochondria I showed over here. This long mitochondria, that's like an example of unperturbed tubular mitochondria. Yeah. Whereas the ones I have, are usually about this size. Yeah. This one's probably, I think this is HeLa cells, and my actual data is also from HeLa cells. Yeah. Also, when you're milling mammalian cells, you always end up, getting a huge. So this whole black area is the nucleus. So you always get a huge nucleus in your lamella, which means that you're also looking at mitochondria that are closer to nucleus, and that's also physiologically different than mitochondria that are on the edges of the cell. The length, yeah, that's the scale bar for this image, probably 4 microns, yeah. I think this is naturally what they look like. They look like long spaghettis that like start at the nucleus and go all the way to the edges. When it's round, it's like 10 to 15 microns, yeah. But when it attaches and it like kind of grows out, it's more like it looks like an eye. I think, yeah, that's like a good example of, they start from the nucleus and kind of radiate out. All right, so, and then I used ArciaX, which is now a plugin in ChimeraX to trace these back. So you import your Dynamo or star file, your Dynamo table or your star file or your coordinate file. It takes in different formats. You open your segmentation and then you assign your maps to each class of particles, and it creates a beautiful visual of where your subtomograms are in the tomogram. So it's pretty easy to use. It's like you open ChimeraX, you type it, run a command, and it will install the plugin, and then you can immediately use it. It's pretty awesome. Oh, ArciaX, ARTIAX. Do you have time? All right, I can probably skip these and open it up for discussion. I just compared the distribution of the different classes between overexpression and control, and then compared it to the ER mitochondria distances for each tomogram. And this was a way to see if these two different kind of analysis that are kind of independent converge and make sense together or not. And I think it does. This is for control. Yeah. So during the process, we also saw some interesting stuff. So usually when people look at contact sites, ER has a negative curvature, which is like a cup. Mitochondria has a positive curvature. But we saw many different examples where it was either flat or mitochondria actually had a negative curvature. So there's some heterogeneity in that. And also we saw some mitochondria nuclear envelope contact sites, which were pretty cool. Yeah, I think that summarizes it. Thanks for my PIs, the great NCITU team, and my collaborators. Thank you so much. Yes.
The segmentations that you showed, were those in like cyber?
Okay, yeah, so let's see. Doesn't matter. So I used deconvoluted isonet tomograms, which gives you a little bit more contrast. Then I used Tomo Segment TV, which basically is a tensor voting algorithm, and it amplifies the signal from membrane. So it basically removes all the background and leaves you just the membrane signal. But there's some noise, and it's not always perfect. So you fix that in Amira, and you also label everything in Amira. And then you can take that to the surface point cloud reconstruction. Yeah, it's a very manual process. So Tomo Segment TV tries to classify your objects based on continuity, but with missing wedge artifacts and just the crazy environment of the cells, you always end up getting like random objects that don't make sense. So whatever is close to each other is one object.
This is the one that gets microtubule.
Your talk. Jack is a scientific programmer and cryo-EM specialist at the EM Data Bank. He works on developing the one-depth system, annotation of entries, and assisting in the management and direction of the EMDB. He had a previous life before the EMDB, working in Peter Rosenthal's group, where he studied influenza A and other cryo-EM and cryotomography work. Something that's special why we're bringing Jack on is a lot of you are creating a lot of Tomo data sets. And the question would be, you're going to publish that. And how can you actually archive that, not only in the EMDB, but EMPIAR? And I think that is sort of a topic where it might be easier now because of Jack. We'll find out pretty soon. So Jack, take it away.
Thank you. Yeah, so hi everyone.
Sorry, we wanted to take a picture of you. I didn't realize.
How do I? If I stop share, will I appear again?
Sorry, if you're on our Slack workspace, you'll see it.
Am I good to go?
Yes, please.
Okay, let me just drag these around here. Hopefully this is coming through okay. Yeah, just shout if anything's not coming through clearly. But hi everyone, I'm Jack. I work at the Electron Microscopy Data Bank, and today I'm going to be giving you a session on archiving electron microscopy data at both the EMDB and EMPIAR. So just to give you an outline of this talk, I'm going to start by introducing the two archives, going through a bit of their history and a bit about what they hold. I'm then going to talk about websites and the validation protocols that we have at the EMDB. I'm not going to spend time telling you how to use a website. You all know how to do that, but we have some things under the hood, including how to do statistical analysis of the entire archive, which you can do in your web browser. And I just want to show you a bit about how to do that, because this can be quite useful for setting the scene and things like this in your presentations. And then finally, I'm going to talk a bit about deposition, both on the side of EMDB and EMPIAR. So without further ado, let's get into introducing the EMDB. So it's the Electron Microscopy Data Bank. It was established back in 2002, and it's now a WWPDB partner alongside PDB-E, RCSB, PDB-J, and BMRB. It is a home for electron microscopy volume maps and tomograms, and all the data is freely and openly available under a CCO license. I've got this bullet point here on all of my slides, which involve an archive. What this basically means is you are free to download and do with the data what you wish. In terms of citing it and things like this, traditionally you would place a picture on your slide like I've done here, and then you'd have the accession code below. If you are putting it in a document, though, you would tend to put the accession code plus the publication associated with that. The two examples I've got here are just a slice from a tomogram of some COVID virions, and then on the right-hand side, this is actually electron crystallography entries at 0.9 angstrom resolution, so you can actually see the individual atoms here. The reason I wanted to put these two up is just to show the scales that we really actually archive across. So we have really all the way from atoms here to viruses. And when I show you a bit about what we have in EMPIAR, you'll see that we archive all the way up to organisms, which of course is something really only electron microscopy can do to be able to collect over all of these scales, which makes it a very exciting technology. On the right-hand side here, I've just got a graph showing the EMDB entries released per year and the cumulative number of entries released throughout history, calculated as a sum each year. We can see that we're increasing in terms of the total number of entries released yearly at a sort of linear rate year on year. Whereas in the cumulative number of entries released, it's actually an exponential function, and we can fit an exponential function to this with an R-squared of about 0.99. So we passed 20,000 entries last year, but actually fitting the curve, we're looking to pass 50,000 entries in about 2025 and 100,000 entries in 2028. Of course, it may not say exponential forever, but considering what we're currently seeing, I do think we will hit that 50,000 figure sometimes in 2025 at least. In terms of the different methodologies we accept into the EMDB archive, I've just got the 5 different methodologies here and an example for each. So we have single particles, so tomography averaging, helical reconstruction, tomography, and electron crystallography. And I've tried to put an interesting example in each of these. So on the side of single particle, we have this gigantic photosystem entry. So you can see the lipid bilayer along the bottom here. And then you've got these two gigantic things coming out of it. And there's a huge number of chains here. We're talking over 200 total PDB chains. The reason this one's interesting is it actually broke the EMDB data out pipeline. We did manage to fix it. It's okay. And if you go to the entry, you'll see the web page is there. Although the 200 chains is an interesting task to load. But I thought this was quite interesting. This was only released a couple of months ago, and actually, we often see single particle as this fully developed method, and everyone's doing it, and it's quite standardized now. But people are still pushing the limits of what you can do, at least on the computational side there. In terms of subtomogram averaging, we have this DNA origami signpost. I think this DNA origami work in general is quite fascinating, but this application of making it a signpost for volume EM techniques to be able to signpost your proteins is something very interesting, and that's something I'm watching quite closely at the minute. On the side of helical reconstruction, we have this tobacco mosaic virus. This was released a couple of months ago, and it's currently the highest resolution helical reconstruction in the archive at 1.85 angstroms, beating out the previous highest resolution helical reconstruction at 1.86 angstroms. But that 0.01 angstroms counts. On the side of tomography, I put in this filamentous virion. The reason I put this here is because it's actually the largest tomogram we have in the archive in terms of total data size. It's 55 gigabytes in size. I wanted to put this here because I think particularly if you've used the one-depth deposition system before, there can be this conception that we can't really handle large files and things like this. Whilst I appreciate sometimes it can be a bit of a struggle to upload very large files through a web interface, we can manage it. And if you are ever having issues depositing your data to us at all, please do get in contact, and we will do everything we can to get your data into the archive. Even if that can mean you sending us Google Drive folders and stuff, and we upload it on our side, we will do everything we can to get your data in. And then finally, on the side of electron crystallography, we have this metarhodopsin 1, which was the first electron crystallography entry deposited to the archive. So that gives you a brief overview of the EMDB and its growth and the type of entries that we see in the EMDB. Now I want to talk to you a bit about the Electron Microscopy Public Image Archive, or EMPIAR. And this is a bit of an interesting archive because it started as an archive that was supposed to take the raw imaging data used to generate the things found in the electron microscopy data bank. So the idea being that you deposited your single particle structure to the EMDB, and then you deposited your raw data EMPIAR. This would allow people to not only validate your EMDB entry, but also potentially take that raw data, use it in new refinement protocols and things like this, and be able to test how well the new refinement protocol is doing relative to the previous, and so on and so forth. It has expanded significantly in terms of the number of methodologies that it accepts now, though, and it also accepts volume EM techniques, X-ray tomography and microscopy, so on and so forth. I'll show a full list later on in the talk. Something that's worth noting on the side of TEM and SEM tomograms, you are expected to deposit a sort of example tomogram to the EMDB, and then you can deposit the rest of your data set to EMPIAR. So the idea being, if you've got a really nice tomogram that you're showing in your paper, you deposit that to the EMDB. But if you've got a data set of 1 to 200 tomograms, let's say, you can deposit the 1 to 200 tomograms to EMPIAR. And it's a lot easier to upload that number of tomograms to EMPIAR in one go than it is to the EMDB. So we recommend doing it like this. In terms of the total number of entries released per year and cumulatively, we can see similar trends to the EMDB, but much smaller numbers. It's not mandatory to deposit to EMPIAR, but we are still seeing quite significant growth at the minute. And the headline figure that we passed last year was 2 petabytes of total data in the archive. And actually, whilst that was a big milestone at the time, I looked recently and we're already at 2.76
Jack,
data. Yes?
can you hear us?
I can, yes.
There's a recurring theme here.
Oh, Jack, we lost you on this end for a second.
Ah, okay, sorry about that.
Yeah,
Am I coming
that's
through?
all right. If you just, you're coming through, but not to the full room. So we're reconnecting to the system that's connected to the full room, and I'll let you know when I'm good.
Ah,
on our audio system.
Check.
No.
Oh yeah, can you hear me again?
Yes. Is that, how's the volume for everyone? Speak a little bit more, Jeff.
What stage did I get to, or when did I cut out, should I say?
You wanted to say something very important about this.
On validation. You got to this slide, and I don't know where you left off.
Okay, I'll start from where I think I was on this slide then.
Okay.
Okay, perfect. So yeah, I would think, if I understand correctly, I was just at the stage of saying that originally this was the home for raw data for EMDB maps, but it's since expanded to cover volume EM techniques and X-ray tomography and microscopy techniques as well. It's worth noting though that you should have to deposit an example tomogram to the EMDB, and then you can deposit the rest of your data to EMPIAR. So the idea being, if you've got some nice tomograms you're showing in your publication, probably put those in the EMDB. And if you have a full data set of 100 to 200 tomograms or something like this, then those can all go to EMPIAR, and it's far easier to deposit those sort of large data sets to EMPIAR in one go. It uses like Globus and Aspera and things that can deal with you losing connection and things like this, which OneDep isn't quite so resilient on. I want to show as well, we've got here, turn off my laser pointer, another example of a FibSEM tomogram. I'm going to butcher the pronunciation of this, but it's a Plataneris domerili. It's a worm, basically. But just to illustrate that in the slide with the EMDB, we had that atomic level electron crystallography entry, and here we're showing full organisms, and we really archive the full breadth of structures in that way. On the right-hand side here, I've got a similar graph to the one I showed on the EMDB, where we've got cumulative number of entries released, as well as the number of entries released annually. And you're seeing sort of similar trends here with a linear increase year on year, but it's much smaller numbers. It's not mandatory to deposit to EMPIAR. You don't have to do it, but if you do it, it will support the field as a whole. The headline figure though last year was that we passed 2 petabytes of total raw and imaging data in the archive, which was quite a huge milestone at the time, but actually I've recently checked it and we're already at 2.76 petabytes of total data. So there's a huge amount of data there. So that gives you a brief overview of the EMDB and EMPIAR. I actually just wanted to stop talking about archives for a second to talk about data reuse and what this data can actually be used for and how it helps the field as a whole. So at this stage, I can actually now fill a PowerPoint slide with machine learning methods that have come out of being able to use this raw data to train machine learning algorithms and things like this. And I'm sure this is not an exhaustive list. These are just the ones that I personally have found. So you've got things like Deep Enhancer, which is post-processing, and that uses all the half maps in the EMDB. Deep Consensus, which is for particle pruning, and we use EMDB and EMPIAR. DRP Net, particle picking, again, this is EMPIAR. cryo-ET, once again, particle picking, and this used both EMDB and EMPIAR. And then Warp and M, which are involved in pre-processing, particle picking, image denoising, and refinement, which also use EMDB and EMPIAR. It's not just about machine learning though. We of course have the more traditional programs like Relion, System, CryoSpark, all of which use data sets in the EMPIAR and deposit their own data sets there as well. And there's other interesting work coming out as well, like Cryo and Crowd Tools, and this paper as well, which is particularly interesting, where they use Deep Enhancer and Side Splitter with Relion. And if you are doing subtomogram averaging and things like this, particularly of membrane-bound proteins, this paper in particular might be of interest to you. I think some interesting results there. Of course, when talking about EMDB, we can't not talk about the protein data bank. So this is the archive for archiving EM, X-ray, and NMR data. I've just put this graph over here, which is the number of entries in the EMDB with fitted models, and currently there's around 14,000. To give you an idea, when I pulled this graph a month or so ago, there were about 26,000 entries in the EMDB, so just over 50% of them have a fitted model. Something worth noting though on the side of PDB is it is a collaboration between PDB, RCSB, and PDBJ, and they all collaborate, and we collaborate with them actually on the side of data in. But they are sort of competitors on the side of data out. So they all have their own websites, and all of their websites host the same data, but they might present that data in slightly different ways. And it is worth going to each of the websites and having a look at how they present the data to see what works best. So just to summarize this part of the talk, in terms of EMDB, EMPIAR, and PDB, it allows the archiving of the full electron microscopy processing workflow. EMPIAR is supporting the development of software and scientists. It's also now an archive where volume EM data can live. The EMDB then includes the refined EM structures and those tomograms that you would tend to find in your publication. And then the PDB, the archive models, X-ray, EM, and NMR. So that gives the overview of the archives that I wanted to talk about today. Now I'm just going to go through the EMDB website and validation. Like I said before, I'm not going to show you how to use a website. You guys know how to use that. I'm just going to highlight some interesting facts about the website and things that I think you guys might be able to reuse in your talks and things going forward. So for those that haven't previously gone to the EMDB website, the link's down here, and when you go there, you'll end up on a page that looks something like this. If you do want to deposit your data, you can click the big red button. If you want to deposit your data to EMPIAR, if you mouse over this deposition thing here, it will drop down a list that you can deposit to EMDB or EMPIAR. As you scroll down the page, you get this Explore EMDB section, and in this case, it's just showing by EM method, and you can see that 81.9% of the archive is single particle, 5.5% tomography, so on and so forth. The reason I want to highlight this is just to let you know that every graph on our website can be interactive. So if you were to mouse over this segment on the website, it will tell you the exact number of entries that the archive has, which are tomography entries. And if you were to click on it, it would return a search result, which would be all the tomography entries in the archive. This button in the top right-hand corner as well can also be clicked to either download the pie chart or download the data behind the pie chart and use that to make charts of your own. I'll show you how to make more specific charts. Also worth noting, we have recent entries here. So this widget allows you to scroll through the recent entries. We can see in this case, in the week that I pulled this, there were 95 new entries. We have a weekly release schedule, the same as the PDB, where we release entries every Wednesday, but those Wednesdays are staged the Thursday before. So if you are depositing to us and you realize that you need your entries released, the application is asking for them released, please do let us know in advance, because these go into staging Thursday, 6 p.m. UK time, before Wednesday. Can't just release them the next day. They have to go through a whole release protocol and pipeline to do data integrity checks, run validation metrics, all of these types of things. The second thing I want to show you about is the EMDB statistics. So there's this big yellow button here, which is EMDB statistics. If you go on this, there's a list of like 100 graphs that represent the EMDB archive in various ways. I've just pulled out one of my favorite ones here, which is the EMDB entry resolution in shells per year. The reason I really like this graph is in the field of EM, we often hear about this resolution. With the EMDB and the data in it, we can actually plot resolution. We can see up until about 2013, the structures tended to be less than 15 angstroms in resolution. There were still higher resolution structures, of course, but they weren't the norm. And actually, we see a very, very rapid increase in high resolution structures after this time, with sub-6 angstrom structures becoming the norm from 2017 onward. We then get sub-4 angstrom structures becoming very common from 2019 onwards, 2018 onwards, becoming over to the archive. And sub-3 angstrom structures are on their way to become sort of 25% of the yearly releases. Actually, I checked, but we're currently seeing an increase in sub-2 angstrom structures as well, which is quite interesting. Not to say, of course, that you can't still get a lot of interesting information from low resolution structures, but we can see that the resolution revolution really hasn't stopped. People are still higher and higher resolution structures. But of course, you might want to look more specifically into the archive and plot things that are specifically interesting to what you guys are doing. And we've recently released this new thing called the Chart Builder. This was released a couple of weeks ago, and it's currently in beta, but actually it's really quite powerful in its current form. So we've decided to release it as is currently, and we're going to make improvements as time goes on. And here I've just plotted 4 lines, which are basically the 4 different structural determination methods here. So every structural determination method except a single particle, because that kind of dwarfs everything else and makes it difficult to analyze what's going on. But we can see from just what I've plotted here, and I've truncated this to 2022 because 2023 is not yet finished. But we can see that we're seeing a rapid increase in subtomogram averaging and tomography entries. Helical reconstruction is also on the rise, but not quite at the rate of the other two. And electron crystallography is keeping along, sort of having a similar entries, at least on average over time. But you can represent this data in a number of different ways. So here it's represented as a line graph of absolute numbers, but we can also represent it as a percentage of the total releases in each year. In this area graph here, and here we can see tomography entries are rising. are on the rise relative to these other entries. Helical reconstructions are staying pretty consistent. Same for electrocrystallography, and in cell tomogram averages, whilst we're seeing an increase in total number, as a proportion of the total releases, they're actually on the decline. So I'm going to show you in a minute a little bit how to make the queries that you can put in here, but then you can go and have a play with this and use the EMDB archive as your data resource for plotting these graphs. As before as well, you can download these graphs using these buttons here, and you can also download the raw data behind these graphs, so you can plot the graphs in whatever software you prefer. Just quickly though, to create those filter queries, we have to be able to run a search. So just a very quick overview of our basic and advanced search. The advanced search, works very much how you'd expect. You build up an advanced search query with various conditions. Our basic search in some ways is actually a little bit more complicated. So here I've written the word ribosome, and if I were to click enter, it would search the archive for the word ribosome in every bit of metadata in every entry in the archive and return a search results list with all of those in. If I want to search something more specific, like ribosome in title, I then have to click one of these things. So this all comes up when you start to write something, and it's all the auto. The other thing I wanted to highlight here is we have something called the EMIX project, which is done by someone called Amula, who's working for us, which is an added annotations project, whereby at the stage of release, we data mine other resources outside of the archive and outside of the data that's been given to us during deposition to add annotations to all of the entries in the archive, which weren't actually provided to us during deposition. And one of those is gene ontology. So we can see here that you can actually search for things like ribosome biogenesis, ribosome binding, and things like this. You don't have to just search specifically for a protein. You can search by protein function and things like this. So if you were to type in membrane, for example, you can search for membrane proteins or viral membrane proteins, things like this. Once you've run your search though, you get a search results page, which looks something like this. So in this case, I've searched for HIV1 being anywhere in the metadata and the structure determination tomography. And this is where your filter query will build up. So if I were to click any of these filter by facets on the left-hand side, it would add additional bits to here, and you'll build up these long queries that you can save and then search again if you want to. But these are also the queries that you would copy and paste into this filter query section on the statistics. And you can plot by a number of things. So you can change the data on Y to resolution over time, EMDB entries over time, so on and so forth. We also have a number of other functions on our search page. So we've got this graph along the top, which shows the total number of entries per year. As I said before, every graph can be interacted with. So if you clicked on any of these points, it would then filter the current search query according to year. So if I clicked here, it would do HIV1 tomograms in 2016, for example. We have the statistics button that just plots some interesting statistical graphs on the current search query. You have the search results down here. If you click here, you'll get taken to the entry page for this structure. And then we have this download button as well that lets you download various things about this entry. So in this case, it's selected JSON metadata. So you'll download all the metadata associated with the entries returned here. But you also have abilities to download that in CSV. And you actually, as well, you can download all of the primary maps in your search results. So if you want to do, I don't know, an investigation into all the ribosomes in the archive, you can get your 1,000 plus results, and you can download those 1,000 maps all at once if you want a large scale data analysis. So that really runs through what I wanted to show you about the EMDB website. It was maybe that statistics builder that I wanted to show you for you there, so you guys can go away and have a go at that at some point and potentially make some interesting graphs that you can use in your presentations. I now want to touch on EMDB validation. So why is validation so important in EMDB? So whilst we are an archive and we have policies and procedures that entries must abide by, and we do try and enforce those as much as we can, we are not the structural biology police. It was decided when we were all set up that the structural biology police really was the community. It was you guys that have knowledge of both the techniques and the structures and biology we're working on. But to facilitate the ability to critically analyze all of these entries, we provide a lot of validation metrics that you guys can go in and have a look at. And we split these into 3 tiers. So we have the validation analysis pages, validation tabs, and the WWPDB reports. So the validation analysis pages, the VA page can be found here. I'm also going to show you in a minute how to get to these pages on an entry by entry basis. And these validation analysis pages are essentially our sandbox. This is where all of the validation metrics that we add start out. It has our most fully fledged sort of series of validation metrics. and then we use this as a place to test all the validation metrics against every entry in the archive and come up with appropriate ways to present it to the end user. This is particularly helpful when we're integrating new validation metrics. So the newest one we integrated was something called Q-score, because the software is not written by us, it's written by other scientists, and then we integrate it into the pipeline. And it is very unlikely that a software developer has had the opportunity to test out their software on 25,000 EM entries. But our pipeline is written in such a way by Reza, who codes our validation analysis pipeline, that these programs can be plugged in and run on all 25,000 entries and then tested on every entry. And we can then work with the software developers to help improve their software and carry out bug fixes. That means you as the community know that the software that you're using has been tested on all the historical EM data, and we then have this data in the archive as well. So once it's all working and once we're happy with the way that we're presenting it, we can then move this to tier 2 of validation. And tier 2 of validation is the validation tab on our entry pages. So that search results page that I previously showed, if you were to have clicked on that entry that was there, you would get taken to the overview page. And then as you go along, you would get taken to the validation page here. And this is tier 2. If you are interested in seeing tier 1 validation analysis, you can click this EMDB validation analysis button here, and that will take you to tier 1 validation analysis for that entry. Tier 2 validation analysis basically contains a smaller subset of the stuff that is visible in tier 1. So this is stuff that's been fully tested, both on the side of it working on all the entries, but also in terms of how we're presenting it to the user. It then undergoes a similar round of review where we decide what's the most useful to the end user and what is seen to be working by the wider EM community, because this is now a place where many more people will visit. And when considered appropriate, we'll move them to tier 3. And tier 3 are the WWPDB validation reports. These are the PDF reports that you get after validation, after annotation, sorry. And these are the things that are then provided to the reviewers of the publication, and they use this to assess the structures that you are depositing. There's a little bit of a lead time between tier 2 and tier 3 because tier 2 is this web-based interface. And as I said before, we try to make it so everything can be interacted with and you can click on things and things like this, where obviously the PDF report is a static image. So we have to sometimes make some alterations to figure out a useful way to display it in the PDF. Also, we are aware that if we make a PDF report too long, people will tend not to fully read it. They'll tend to just skim it or not even look at it at all. So space comes at a premium. So we have to just pick the stuff which really concisely conveys the message that is important to be conveyed for the analysis and validation of that structure. In terms of some examples of the validation metrics that we present, I've just got a couple of examples here. So we have some qualitative metrics. Idea being, if you're going to an entry for the first time, you may not have looked at the map yet. So we have some different slice views. We also have in the case of single particle subatomic averaging, helical reconstruction, some views of the surface view as well. This is rendered at the user provided contour level. So if you are depositing subatomic averages and you provide a contour level, you're asked to provide a contour level, please provide one which sort of is the correct contour level for the display of your protein. We do use that when we render them here. We also have some quantitative metrics as well. So these are quite recognizable, the FSC curve and the rotation average power spectra. Of course, these aren't all relevant to tomography. These are things that we have for subatomic averaging. In terms of tomography, we have a whole bunch, this is tier 1, I should say. We have a whole bunch of different slices. So we're currently actually investigating whether there's a technique we can use to pull out in a repeatable way the nicest slice of a tomogram for viewing. So we have a whole bunch of slices in tier 1 that you can view here. And then on the side of quantitative, we actually only have the voxel value distribution at the moment. If you do have any ideas for more quantitative measures, we can add to tomography validation. We are all ears, by the way, because we are aware that we have fewer quantitative metrics that can be used for validation of tomography methods. In terms of the full list of validation metrics that we provide, I've shown a whole bunch of slices just there and large variant slices and things like this. We do also show the EM map with masks. It's worth noting that if you do deposit a segmentation, you can deposit a segmentation as a mask to the EMDB, and we will then present that on top of the tomography map as well. We have a whole bunch of volume graphs. I've just shown a number of those. And then if you also include a model in your deposition, if you are depositing a subtomogram average, we also provide a bunch of map model fit analysis as well. And Q-score, which is inferior, is a map analysis but it requires the model as well. So that really covers what I wanted to talk about in terms of EMDB validation. Now let's get on to talking about deposition. So I'm going to cover two aspects here. I'm going to do EMDB deposition, and then I'm going to talk about EMPIAR deposition. It is worth noting that I work for the EMDB, I don't work for EMPIAR, but I've sat down with the guys at EMPIAR and put together some slides to show you today, which will hopefully be helpful when you come to deposit there as well. So the first thing we have to talk about when we're talking about deposition to the EMDB is OneDep. So OneDep is a unified deposition system for EM, X-ray, and NMR. You remember from earlier that I said the EM maps go to the EMDB and the coordinate models go to the PDB. You, as a user, do not have to worry about that. You deposit them to the same place, and we deal with that data route. We have 3 sites throughout the globe. So we've got PDBJ in Japan, we've got PDB and EMDB where I work in the UK, and then RCSB in the US. The way that the OneDep system is set up is that depending on where you deposit in the world, which country you're in, will depend on where your data goes for the lifetime of annotation. So even though the EMDB is in the UK and we are the archive holders of the electron microscopy data bank, if you were to deposit your data in the US, it will go to RCSB for annotation and getting your validation reports. At time of release, it will then be synced across to the EMDB where we will release it on our website. So this is the way that we currently function. So all EM entries are annotated by all 3 sites. In terms of the type of deposition as well, I've gone through the different methodologies we accept, but that doesn't quite tell the whole story. We actually accept 3 different deposition types. So we can have map only, model only, or map plus model depositions. In terms of map only depositions, this will tend to be the deposition type you use when you're depositing a tomogram, but you can also deposit anything as a map only deposition. It's the experimental data. We don't have any rules on that. It's worth noting that in theory, left over from legacy entries, it is possible to deposit a tomogram as a map model deposition. This has been done in the past, but generally I think I would recommend that they come in as map only depositions. I don't know how the community currently feels about building models into tomograms with how it is at the minute. We also allow model-only depositions, but it's worth noting that no model can exist in the PDB without an associated primary data. And when we're talking about electron microscopy, the associated primary data is a primary map. So the idea behind this is that if you do deposit a high-resolution primary map and you later want to deposit a model, you can do that without having to start a completely new deposition from scratch, which is a map model deposition. You can just start a model-only deposition and associate it to the previously deposited map, and that will all link up and will be validated together. And I'll show you a little bit about how to do that in a minute. And then finally, we have our most common type of deposition, which is the map plus model deposition. And this is where you deposit your map and model at the same time. You provide all the metadata at the same time. It goes for annotation together. You get the validation report, and we deal with it on the side of data out and put everything in the correct place, and you don't have to worry too much about that. Okay, so you know now the methodologies and the different deposition types, and you have an idea of what you want to deposit, and then you're going to have to start thinking about depositing. So I'm not going to go through with you how to fully fill out a web interface, but I'm just going to highlight a few bits that is worth knowing about for helping speed things along and setting up different deposition types. So when you come to deposit your data to OneDep, you'll come to this page here, which is the OneDep homepage. In this case, I've already selected a country. So here I've selected United States, and you can see that's highlighted in red here. So this would go to RCSB. You're then going to select the type of experiment you do. So I've got that zoomed in on the right-hand side here. And you can see you've got the 4 sub-methods of electron microscopy, and then electron crystallography exists just by itself over here. When you select one of these, so in this case, I selected subtomogram averaging, you'll also have extra options appear depending on what you've selected. And you can do various things with these. So are you depositing coordinates with this submission? If you selected no to this, that would initiate a map-only deposition. Has the associated map been deposited previously? If you selected yes to this, that would initiate a model-only deposition. And you would then have to provide the EMDB accession code just here of the associated primary map before you can proceed. And finally, is this a composite map deposition? I haven't gone through this today because this is a tomography course and I wasn't sure how relevant it would be. But if you are playing around with composite maps and doing subtomogram averaging, you would click yes here and some red text would appear underneath that would explain what you have to do. I do recommend reading this thoroughly and also going to our EMDB policies if you are doing composite map deposition and reading policy number 1.3. This will explain exactly what to do. Basically, you will need to upload multiple entries with different maps, like your focused refinements and things like this. And they will have to be their own depositions so that they will get validated individually. The next thing I want to highlight on this page is something which we added last week, which is this sign in with ORCID button. If you previously deposited to the EMDB or PDB, you will know that when you click start deposition on this page, you get sent a 10-digit deposition ID with no other metadata associated with it. I think it comes with your password as well. And then you have to put that into an Excel spreadsheet to keep track of everything that you're doing. It can quickly get out of hand and trying to figure out, what's got messages you need to reply to and things like this. We're trying to alleviate this problem, and the first step to alleviating this is sign in with ORCID. If you sign in with ORCID, you will get this table here. And if you have previously entered your ORCID in any of your depositions before we added this, your entries will already appear here when you sign in. And you can see you will have all your deposition IDs here, all of your entry IDs, entry titles, so on and so forth. This is the first version of this. As I say, it was only released a week or two ago. We are planning to add new functionality to this as well, such as reordering the columns and also being able to search these as well. One thing I did just quickly forget to say, sorry, on this slide that I want to quickly circle back to is this is called internally the brain page, because this is where you set up your deposition. And when you start your deposition, it will set up your metadata items permanently for that deposition. So please do be very careful when you set this page up, because there's no going back when you click start deposition. And we do not too commonly, but on occasion, have people clicking these buttons a bit too quickly, and they want to submit a subtomogram average, but they accidentally click tomography, and they don't notice until they get through to deposition, to annotation, and then we have to say, I'm sorry, you clicked the wrong button. You're going to have to go back and restart this deposition. I am currently working on a machine learning project, which will hopefully highlight to the user that they've clicked a particular methodology, but uploaded a map of a different methodology, but that is still in the works. So for the time being, you just have to be quite careful when depositing that. So you started your deposition, you've signed in with ORCID, so your dep ID has appeared here somewhere. You can then click on your deposition ID and you'll come through to the file upload page. File upload basically involves what you'd expect. You choose your files, you upload your files, you fill in a big web form, so on and so forth. There is a lot of metadata to provide though. So to make your life easier, if you have previously deposited an entry, you can use this based on previous WWPDB deposition option, sign in with your dep ID and password, and then tick the relevant boxes that you want to pull across. I highly recommend doing this. It will make your life so much easier. The only caveat to this, and is worth remembering, if you're doing sort of 10 entries at once, you'll have to fill out one entry completely, and then you can pull that information across for the other 9 entries. If you actually want to pull across information from a previous publications deposition or something like this that you did a year or 2 ago, due to the way the system currently works, and we're aware of this, but I'm going to tell you the workaround, it needs the pickle files under the hood to exist for the previous deposition. What this means in plain English is those get scrubbed after 12 months. So you'll need to sign out this deposition, go to the deposition you want to pull from, sign into that so it regenerates all the files, come back to this deposition session, and then run this, and it will pull everything across. If you don't do that beforehand, it will allow you to still fill out the deposition ID and password and all of this, but it won't actually pull any information across because the files just aren't there. If you do want to do this, you need to do this during the initial file upload step. This option will disappear once you've done the initial file upload step. Once you've completed that though, you'll then be given the full deposition interface to fill in. So this has got all of the files uploaded here, and then I've just got an example of what it looks like when it's completely filled in. So each of these is its own page. It's fairly self-explanatory. I'm not going to go through this with you today. You just go and click on each of these and then fill in each page in turn, providing the appropriate metadata. One thing I do want to highlight though is whilst I did just show you an example of one way to pull data across, we have also recently added another way to import data, which is using the MMSIF file format. So MMSIF file format is replacing PDB file format, but it is more than just a model file format. It is also a metadata file format. So we have recently added the ability for map-only entries for you to provide MMSIF metadata files. I appreciate you might not be familiar with how to generate an MMSIF file. What I would recommend is either completing one of your depositions and submitting it and then allowing it to go through annotation, When annotation is complete, you will receive things like the validation report, but you will also receive an MMSIF file of your entry. And then if you open that MMSIF file in a text editor and just Control F for anything that you want to pull across, let's say you want to pull across microscopy information, Control F cryos, that will probably take you straight to the MMSIF category that you're going to want to copy across to a new file to embed that metadata. It will be very obvious what kind of information you need to copy across to a new file. Just save that all as an MMSIF file when you've copied across all the categories you want, upload it as the MMSIF metadata file, and you can pretty much completely fill a deposition interface from your MMSIF file. There are very few things that won't be a green tick here if you use it to its fullest. Another way to get an MMSIF file is we currently don't provide them on the EMDB, but on the PDB we do. So if you find an EM entry and then you go to its related PDB structure in the protein data bank, you can download an MMSIF file from there. And whilst that will have all the atom information as well, it will also have all the metadata information. So you can still search for, EM imaging, for example, and find what you need to fill in there. There is also a website with the dictionary. You just Google MMSIF dictionary and you'll be able to find a lot of the things there. So I just wanted to finish EMDB deposition by talking about some tips and tricks. So as I said before, the MMSIF file is a metadata file. I appreciate this is more of an advanced thing in being able to do this, but I do recommend having a bit of a play with MMSIF files if you can download one and trying to figure out how to exactly pull across your metadata there, because it will just make your life a lot easier when particularly when doing multiple depositions over and over again. You can just quickly fill out all the dep IDs. The second one here is one that is more when you've got a coordinate model, but you can also provide sequences for map-only entries, including tomograms, if it's got an entity in it that you want to provide the sequence for. It's worth noting that the sequence should be the full sequence in the sample, including any unmodeled regions. So often when people deposit, they see that they need to provide a sequence, and they provide the sequence that they've modeled. Actually, you want to provide the full sequence because we're going to match this to the UniProt ID, and that allows us to accurately match that. Half maps are now mandatory for the appropriate techniques. So that's subtomal averaging, helical reconstruction, single particle. These must be unmasked, unfiltered, and unsharpened raw maps. What this basically means is they must be the maps that come straight out of your refinement process. Defocus values must be in nanometers, and positive values refer to defocus. I put this in here because I know on microscopes they are negative, referring to defocus, and often given in microns. I didn't make the dictionary. This is how it was when I arrived. Just so you're aware, positive values refer to defocus, and they must be in nanometers. I have recently added warnings to the deposition interface if your values look like they're in microns or they're negative. So it should highlight it in red or yellow when you type in something that doesn't look quite right, and it will let you know that as well. Maps and models should all be present in the same coordinate space and overlaid correctly. Basically, we allow you to do anything to your primary maps. This includes things like cropping them. But if you were to deposit your primary maps and half maps, they should still overlay correctly. So if you crop your primary map, for example, make sure to shift the origin in such a way that it still correctly aligns with your half maps. Same in tomography. If you are depositing a tomogram and you're also depositing segmentations, please make sure they overlay correctly so that we can have them all overlay correctly in the validation reports. And finally, I mentioned this one previously, when depositing volume EM data, make use of EMPIAR. You don't have to deposit all of your tomograms to the EMDB. You can deposit just a single example tomogram, and then you can deposit the rest of your tomograms to EMPIAR. And this is a much easier process in many ways. So with that, let's talk a little bit about the EMPIAR deposition process. So as I said before, I don't work for EMPIAR. I'll hopefully be able to answer any questions you do have, but I've worked with the EMPIAR team to produce some slides here today to present to you. So the EMPIAR deposition system, step by step, it is far easier than the EMDB. It's far less structured, and it's really designed to be a bit more freeform so it can handle new methodologies and things coming in as well. So in 6 steps here, it really is quite a simple solution. So you register as a new user on the EMPIAR's deposition system. That's easy enough. Sign in, create your new deposition, complete the deposition overview page. I'm going to show you a quick example of that in a minute. Number 4 here, this isn't something you have to do, but I just wanted to highlight that you do have the ability to do this. If you created a bunch of EMPIAR depositions during your postdoc or anything like this, and you want to transfer these to your PI when you leave the lab, you do have the ability to do that right in the EMPIAR login system. You then are going to upload your EMPIAR data. So EMPIAR supports Globus and Aspera. It does also support FTP. If you have any issues with these, you can contact the EMPIAR team and they will help you with that. They don't recommend it because it doesn't quite have the ability of Globus and Aspera to deal with things like losing connection and things like this. But it is possible if you need to go that route. And then finally, you complete the associated image sets with data page and submit. I'm not going to show you this. This is relatively obvious, just filling out forms and things like this. One thing I do want to highlight though, and I should really say this for all the archives that I've talked about, we all have policies and procedures and you can go and read them all. I appreciate on the side of WWPDB, it is a very long convoluted document. EMDB is a little bit cleaner and tidier, I think. But EMPIAR really wins this prize in terms of how easy it is to read. And I really do recommend going and reading these because it is so unstructured, it doesn't really have many rules. And the policies really are about, what we currently accept and things like this. And it's actually a very quite short and concise document. And it contains information like the accepted file types and the supported methods. So I would highly recommend going and reading this. In terms of the supported methods, I've just sort of copy and pasted them here, but actually this isn't the full bit of information. There's more information in the policies document, but you can see just how many different methodologies EMPIAR now accepts. I should note as well, whilst we do have a series of accepted file types, the whole point of EMPIAR is it is supposed to be reasonably flexible and things like this for new data processes coming out. So we do accept more than these file types. The general rule is that we have to be able to open the file to check it. So if you are doing any work and the file type that you're using is not in the list of accepted file types, please just communicate that to the EMPIAR team, and they will then work with you to basically get these files into the archive, making sure that they can open them to check them and these types of things. So don't worry if it's missing, just communicate with them and they'll be able to help you out. In terms of starting an EMPIAR deposition, you can either go to the EMDB webpage where I showed you before, and you can start the deposition there, or you can go to the EMPIAR webpage as well, and you can choose the EMPIAR deposition system. I haven't had time to go through the segmentation or specimen preparation widget in this talk, but the specimen preparation widget in particular, if you are doing lots of volume EM work, I highly recommend going and have a look at this as sort of an online lab book to keep all of your specimen preparation protocols in one place, and also they can then be easily shared and things like this. So I would recommend going and having a look at that at some point. But for the purpose of what we're talking about here, you would go through the EMPIAR deposition system, you would sign in, you would start a new deposition, and then you would travel to the EMPIAR deposition overview page. So this is what the overview page looks like. The stuff in grey and white is optional, the stuff in orange is mandatory. It's worth noting that for everything that is optional, it does have to be filled with something, so you have to fill it with NA, so you just have to click NA here along all of these if you can't fill it out. If you do have a citation information that you can just put in your PubMed ID or DOI and pull that across without actually having to type everything in. The bits that I really wanted to highlight here though is there is a really fully fledged deposition manual. So if you get stuck, you can go there. There is also this help desk button. This help desk actually goes through to both the EMPIAR developers and the annotators. So regardless of the question, how technical it is or anything like this, there will always be someone on that desk that can help you out. Once you've done all of that though, you want to upload your data. As I said before, we support Aspera and Globus. We also support FTP, but don't recommend it. If you haven't used these clients before, don't worry about that too much. There is fully fledged guides on how to do this. And as I said before, you can contact the team if you're having any trouble, and they have quite a lot of experience with troubleshooting Globus and Aspera. So if you are having any problems, please just contact them. Just to give an overview then of EMPIAR deposition. So start by gathering information. This is really going and looking at that policies document that I said before to make sure the data types and things like this are accepted. Organize your data. So the way that you organize your data on your system in the file structure that is organized will be the organization that is then presented on the EMPIAR website. So make sure that you've got everything in their own folders as in where you want them. If you want each tomogram in their own folder, for example, put them each in their own folder and label those folders appropriately. If you are uploading, raw data as well for subtomal averaging or single particle, please subdivide the directories possessing more than 10,000 files. This is just because it takes a very long time to upload folders with lots and lots of little files in them. And the Globus and Aspera programs, they can handle losing connection, but if they lose connection whilst uploading a directory, that can cause some issues and slow things down. So try and subdivide those into 10,000 file folders if possible. Set up your data transfer programs. So this is just a matter of downloading the client of choice. I think Globus is probably the preferred software, but Aspera is also fine. And then finally, get on with depositing your data, which is what we've just gone through there. So with that, I'll just put this last slide up here. So if you are interested in going to any of these things that I've talked about today, there's a QR code here and the link here. I'm going to share a PDF of these slides as well with Ed, so you can get these later as well if you want. The statistics builder can be found here if you're interested in having a go at that. And also just to say that this talk is sort of, I give these talks quite often, and we do ask for feedback on each of these talks so that we can optimize them over time. And the resulting presentations we give are a result of the feedback you provide mixed with the specifics of required for that talk. So this is just a link to a Google form, and we'd really appreciate it if you just took a minute or two to fill that out as well and let us know what we did well and what we could improve and things like this. So with that, I'm happy to take any questions, and thank you for listening. You're muted on your end if anyone's talking.
Can you hear me now?
Yes, I can hear you now.
Okay, so I imagine the community downloads a lot more than we upload. So if the community uploads 1 gig of data to the EMDB, what's the relative ratio that the community downloads?
That's a very good question, not one I actually know the answer to. I think we'd have to check our Google Analytics page. I think you're right in some ways. I think for interesting structures, the amount of data, total data downloaded will be much higher. But the unfortunate truth is I think a lot of structures also are left untouched. So I don't know the exact ratios, but yeah, I imagine more data is downloaded than uploaded.
And then you showed a graph of exponential growth. So if you have a couple petabytes right now, are you ready for 4 to 5 petabytes in the next couple of years?
Yeah, so this will be interesting. For the time being, we're okay. So at EBI, we have all of the other databases as well, AlphaFold DB, UniProt, so on and so forth. And actually, whilst 3 petabytes sounds like a lot of data, surprisingly, the genomics databases, we're still sort of small by actually. But if it continues at its current rate, there will be a time, I suspect, where we need to assess other data storage solutions and things like this. You're quite right. But for the time being, we're okay.
Are there any efforts to make like AI get metadata from some of these uploads? Because there's like so many fields when you're uploading data, and there's this energy barrier when someone doesn't understand the drop down. Like there's 10 options, and sometimes it's not even like you go look in the header and you're like, it says it's a half bit. That's not a choice. And so there are some things that will auto populate, and those are great. And so I think if there was something along that lines or examples that people could look at, because each of these programs only write a certain type of file for the most part. And so all of this metadata could be captured, but there's activation barriers from users who already don't want to do this because of how many files there are. And if it's optional, they don't know what it is. They're going to skip it. They're not going to figure out what it is.
That's an excellent point to bring up, and you're not the first person to bring that up actually in the talks I've given this year. So I think this is referring specifically to EMPIAR deposition, where you have to select the data type during upload, and I have fed this back already to the EMPIAR team that if possible, this should be the type of thing that's just scraped from the header of the files. There is a slight issue where different file formats can be in the same deposition, and we'll have to deal with that. But I think so far, because EMPIAR is non-mandatory and has mainly had sort of very experienced EM scientists being the ones that deposit, actually software developers as well, that know this information, we haven't really had that feedback, so haven't made the change. But as it's now opening up to more and more people depositing, we have noted that there are some rate limiting steps there that actually are inhibitory in a way to getting people to deposit, and we are looking at improving that and being able to scrape it directly from the file. Yeah, absolutely.
Well, even with the EMDB, like maybe a user should know what type of refinement they're doing and rely on, but they don't. And so if you put if you say you're using rely on and what job you're doing, if it auto-populated that information would be there. Instead, they're just going to skip it instead of trying to figure out what it is. Or you can even put the pressure on rely on CryoSpark to make a little disclaimer. These are the types of things that these do, so the information is out there. You know, like it doesn't have to be your job. It's just I get tired of answering the same question over and over again.
So yeah, no, I can appreciate that. So in some ways, this comes back to that EMPIAR metadata file that I talked about, and maybe I can talk a little bit about that we are on other things that will sort of handle this, where you might imagine, you know, there are software packages out there which sort of give you an overview of the refinement protocol that you're doing, and all of the programs are imported into those software. I won't name any in particular because I don't know what I can and can't say at this stage. But there are people working out there to basically capture all the metadata as you go through and auto-generate an EMDB deposition for you right at the end from everything that you've been doing. So I think being able to, the way that the OneDep system is set up, particularly the deposition interface, having these links would be nice, but then we on the side of EMDB and OneDep would have to maintain those links, and if Rely on ever updated to do different refinement protocols and have different things, we would then have to make sure we very quickly update those so that we support it. So having this handled on the side of the refinement protocols and actually producing these metadata files that will also fill in things, I think that's the direction we're trying to go here to make everyone's life a bit easier on that side.
Cool.
Hey Jack, nice talk. I just want to build on the last question, because especially for EMDB, there are more and more newcomers in the EM field, and then I think some of the metadata we actually don't know what they are, and they are not. We really don't need to know. Like for example, to this day, I don't know what is a assembly in EMDB. Every time we deposit, we got away with it by either leave it blank or select whichever option allow us to keep advancing, and then eventually it uploads, and then whoever downloads thinks our data is okay. So that functions. But on the other hand, like if we do really want to learn something by downloading the EMPIAR data, we actually need more information. Like in my opinion, you actually need a little bit more mandatory terms when you upload to EMPIAR to make sure people can actually deal with it. So is there any plans to implement these, or even you can give certain data sets some kind of label that people can actually reproduce the same result?
Yeah, I guess two parts to this question. You mentioned assemblies there. That's really a holdover from when OneDep was developed for X-ray, and of course with X-ray you have your repeating unit, and that might be biological unit. The assembly ends up being whatever the biological unit is. I can say when we annotate EM entries, the assembly is just what you give us. It's just what you've built. You don't really need to say much on that. You can say, for example, if you've got a protein that's a homotrimer, let us know it's a homotrimer, and we can double check that. But yeah, it is what you've built, unless you provide matrices. So that's a classic example would be helical reconstructions, where people deposit a single subunit and then provide matrices to build the whole helical arrangement. And we will accommodate that, and we will put that in as an assembly in the MMSIF file, which means that when you open it in Chimera, it will open as just one of the monomers, and then you can click the assembly button and it will build the entire helical reconstruction. So that's what that's used for. But I appreciate, yeah, now you said that, I will feed that back, because I do agree that's more of an X-ray term than an EM term in many ways. On the side of EMPIAR metadata, so between the EMDB and EMPIAR, we don't want to duplicate too much metadata. We don't want people having to enter things twice, but also we don't want redundancy between the archives. There is a slight issue though, in that EMPIAR is not yet an affiliated archive of www.pdb. That is planned, but it's not yet there. And until it's affiliated, we can't share the metadata between the archives. That's just the rules of having to keep things confidential and in each of the archives. So the idea being that we would hope we would collect enough metadata on the side of EMDB, and I think in many ways we already do, although not all of it's mandatory, and we do need to review that, to give you all the information you need for how that reconstruction was created. And then you can look at the EMPIAR entry and hopefully make use of that. There are, of course, certain things that depositors don't have to provide, such as particle picking positions and things like this, which may be of interest. But in theory, there should be enough information between the EMDB and EMPIAR to regenerate, at least in some way, the data that's in EMPIAR. But yeah, I can feed back. Out of interest, is there any particular methodology that you've noticed that there's not quite enough information there for? That would be useful to feedback.
Yeah, because this is a cryo-ET workshop, and I was trying to learn cryo-ET reconstructions, tomogram reconstruction. I noticed, at least for some of the tutorial I learned, like at least you have to provide some kind of rough rotation of the axis, or like size of the fiducials if they do have them. Obviously they don't provide this in the publication, and then you almost always have to ask the author about such details.
Okay, this is good to know. So yeah, my background's cryo-ET as well, so no, I can appreciate having to have that metadata. So yes, I can feed that back to the EMPIAR team as stuff that needs to be considered as mandatory. Of course, there is the other side to EMPIAR as well, where now it accepts so many different methodologies. Someone, I think, does need to. We haven't really had the chance yet for someone to go through and determine exactly what should be mandatory and what should be optional for each of these methodologies. So yeah, this is something I can definitely feed back as requiring review. Thank you.
Okay, Jack, thanks so much.
Thank you. Nice to meet you all.
Okay.
Bye.
Have a good day. You can see some of those.
Thanks, Jack.
Thanks, bye.
Okay. We'll send you a couple of pictures, and do send a copy of your slides. And we wish you were here.
Yeah, sorry I couldn't be there today, but no, thank you.